My name is Moses Obiri. I believe we met yesterday, and today I'm giving this talk on missing data and imputation on behalf of Dr. Jen Hackett, and who cannot be here today. And Dr. Jen Hackett is also a data scientist, and our team lead for the statistical decision support team within the National Security Directorate here in the lab. Her technical work focuses on research and development of statistical method to further the lab's strategic capabilities in areas such as nuclear decommissioning, emergency response, risk, and then optimization modeling. She holds a PhD and master's degree in statistics from Iowa State University, and also a bachelor in mathematics from New Mexico Institute of Mining and Technology. And so she prepared this presentation, but she cannot be here today, so I'm doing it on her behalf. And this presentation is on the common issue of missing data, and one way of handling missing data, which is using data imputation methods. And so this is the outline for the talk. We'll go over what missing data is and look at the nature of missing data in our data set. And then we'll look at the implication of inference based on missing values in our data set. And then look at one way of handling missingness in our data set, that is data imputation. So we'll look at some imputation methods and some consideration that you need to make when choosing which imputation method to use, and then look at implications of using imputed data for inference, and then also tackle the issue of single imputation versus multiple imputation. So single imputation is when the inference is based on one imputed data set, and multiple imputation is when your inference is based on multiple imputed data set. So what is missing data? Missing data is when you don't have data stored for certain variables or participants in your data set. And there can be numerous reasons why you run into missing data issues. One common example is data entry errors, where you can just have human error. So that will result in missing data. If it's a survey data, it can result from people refusing to answer certain specific questions. And then at times, the missingness can also even be related to the type of measurement that you are trying to do, that you cannot make certain measurement below or above a certain threshold. So data censoring is also a common way of getting missing data. And so one way of handling missing data is through data imputation. And so imputation simply means that you are filling in the missingness in your data set. And so there are numerous methods that can be used for data imputation. And these methods depends on the type and the nature of missingness in your data set. And overall, multiple imputation is preferred to single imputation, right? Single imputation is when you impute your data once, and then your analysis is based on that one imputed data. And then multiple imputation is when you impute your data multiple times. So you can impute your data m times, and then you analyze all m data set, and then you sort of average result across your m data set, and then you make inference based on the average, the pooled estimate from your M analysis. And so because multiple imputation account for the uncertainty in using your imputation method, it is generally preferred to single imputation, because your analysis are more likely to be more accurate and more capture the information that you are trying to capture. And so this is an example of a data set with missingness in it. So this is survey data where respondents were asked to answer questions related to their level of education, ranging from 1 to 4. So here 1 means that the person has a lower level of education, and 4 means that the person has a higher level of education. They were also asked to answer questions relating to the number of hours they work in a year, the number of months they work in a year, their earnings in dollars, in thousands of dollars here, their retirement interest, and then whether they receive assistance or not. And so here, each row here would represent information from 1 person, and the missing data here is represented by either blank cell with the highlighted yellow cells here, or with NS. So often, depending on the software that you are using, the missing data will either appear as NA or N/A. And so broadly, the nature of missingness falls under 3 categories, right? The first one is when the missingness is completely at random. And this occurs when the missingness is independent of observed and unobserved variables in your data set. And so in this case, the probability of missingness will be the same for all units in your data set, right? So for example, for the survey data set, if for each subject, we decide to collect the earning data by rolling a die and then refusing to answer if a 6 shows up. So in that case, the probability of getting a 6 is the same for all subjects. And so the type of missingness that will occur in the earning variable will then be completely at random. And under this situation, the sample that you get is unbiased. And so if you even ignore the missingness in your data set, the resulting sample is still representative of the population. And so any analysis based on the sample will also get a sort of unbiased estimate. The only issue is that because ignoring the missing values will lead you to get a smaller sample size, you get less power. I think you heard a lot about power in the previous presentation. So you get less power, and then which means that even though your estimates will be unbiased, they will be sort of less precise. The second form of missingness is when it is missing at random. And missing at random is where the missingness is related to some variables in your data set. And so under this scenario, the probability of missingness depends on available information in your data set. And so for the survey data, again, if the probability of missingness in the earning variable, for example, decreases at higher education level, which means that people with higher education level are more likely to answer questions related to their earnings, then that type of missingness is missing at random. And so when you have missing at random, analyzing the complete cases may or not may produce unbiased results depending on how you analyze them. If you are able to account for all the relevant variables in the data set that are related to your missingness, then you can produce unbiased results. But if you are not able to do that, then the result you get may likely be biased, because the sample, ignoring the missing values, will lead to your sample which will not be representative of the population. The last form of missingness is when you have missingness not at random. And this type of missingness is related to either unobserved variables in your data set, or or the missing value that you are trying to the variable that you are trying to use itself. So this is typically common for sense string values, whether left or right sense string. And so for the survey data, again, if, for example, the probability of missingness in earnings is related to eye color, we don't have eye color in the data set. And so that type of missingness in earnings will be missing, not at random. And another example is if the probability of missingness is also decreases as the earnings of the participants increase, then because the missingness is related to earnings itself, then that type of missingness is also missing, not at random. And so typically the type of imputation method and analysis you do will depend on whether your missingness falls under any of these three categories. And so with missing data, one way of dealing with it is sort of getting rid of the missingness in your data set. And doing this will depend on so many factors. One, it will depend on your domain knowledge. It also depends on your sample size, for example, and then the proportion of missingness in your data set. So if you have the missingness is, let's say, 10 or 15%, then you can get away with using the complete case route where you get rid of the missing values in your data set, right? But in that case, even the analysis you do also depend on whether the missingness comes from, whether it's missing at random, whether it's missing completely at random, or whether it's not missing at random, like I just outlined in the earlier slide. There's also the available case route where you get rid of some missing values in specific variables based on your research question. Again, this also suffers the same fate as using the complete case route, where your analysis may be biased or unbiased depending on the nature of missingness that you are dealing with, and other factors like sample size and the proportion of missingness in your data set. If it's a survey data, there are ways of also using non-response weighting methods where you use weighting methods to account for high proportions of missing or completeness from specific subgroups in your population. And so if you are aware that maybe a certain group represents maybe say 50 or 60% in your population, then if you get low response rate from that group, you can then weight responses from that group in a way that will make your data more representative of the population. But the common way of dealing with missing data is using imputation methods. And with imputation methods, we have two broad forms of it. We have imputing the data set before you model it, and then using imputation whilst you are modeling the data set. For today, we will look at just imputing the data set before modeling. And some of these methods that you see here are some of the common methods that we use to impute the data before you model it. And some of these methods are using the overall mean of the variable, using the means of the subpopulations in the data set. And you can also use regression models to also impute your missing values. And there are other methods like matching and hot deck approaches that you can also use. You can also do indicator imputation, where if your variable is a categorical variable, then you then add a new level for the missing variable so that you get an extra level for your categorical variable. You can then also use, if it's a continuous variable, then you can then use an indicator variable where if you observe the value, you assign it a value, say 1, And if you didn't observe any value, then you assign a value that says 0 to it. And then there's also random imputation that can be used for single or multiple variables, where you draw multiple values from a probability distribution. If you are using a regression model, you can use the predicted value and then the standard error of prediction to draw values from a known distribution. Often the known distribution that is used here is a normal sort of distribution. So this is an example of using the overall mean to impute values for the missing values for the survey data that we looked at. So to do this for the earnings variable, you just find the average of the observed values, and then you replace the missing values with this average. And so you can do the same for the other variables. For the retirement, you do the same thing. You find the average of your observed values, and then you replace it with you replace the missing values with this average. At times, using the overall mean may lead to results which are not accurate. So, for example, here, if your missingness, or if the variable that you are trying to impute the missingness for is related, if it has sort of a relation with other variable in your data set. So here you can see that the mean log earnings increase as the level of education increases, right? So in this case, instead of using the overall mean, you can use the subgroups here. So you can impute the missing earning value for an individual, depending on which education level that they are in. So I think this type of missingness would be missing at random, where the missingness in earnings is related to whether the level of education that the person has. And so here, using an overall mean will not be the right approach here. And so using the mean of the subpopulation, and here the subpopulation is levels of education, will be the right way to go. And then there's also regression imputation, where depending on the type of missingness that you have in your data set, you can then use a regression model to sort of model your variable as a function of the other variables in your data set. And then you can use predictions from your model to then sort of replace the missing values in your data set. So in our survey data, we can model earnings as a function of the other variables in the data set. And then we can then sort of make inference by imputing the missing values using our regression model. With regression model, we have to be careful to make sure that the prediction that we are doing is accurate. And I think you had an extensive talk about looking at assumptions such as normality and other stuff to make sure that your regression model is accurate. One way of doing this is that if you see a scenario where you have some potential outliers, or that your regression model do not follow a normal distribution, you can then apply different transformation like square root or log transformation. So in this case, we applied a square root transformation to get a better model so that our imputation will be more accurate. For the case of potential outliers, you can use top coding, where all values above a certain threshold, that is the top code, are set to that threshold. And so that this will then prevent extreme values or outliers from having an unduly large influence on your regression result. So one good thing about using the regression model approach is that you can you can use it, you can do random imputation from it. And so with random imputation, you use the predicted value and the prediction error to sort of draw values from a normal distribution. And you do this to account for the uncertainty in using your model to sort of estimate the missingness in your data set. So the example that you have here is that you can either, when you use your predicted value to replace your missing value, you are sort of getting a straight line. And so you are sort of not accounting for any uncertainty in your model, but then you can then use random imputation where you combine the predicted value and then the prediction standard error to then draw multiple values from a known distribution, in this case, from a normal distribution. And then random imputation is one common way of sort of even doing multiple imputation where you draw multiple values to replace the missing value that you want to sort of replace. And so the imputation methods that we've talked about are sort of some of the simple methods that you can use. You can also use more complex approaches depending on your domain knowledge. You can, for example, use a multi-stage imputation method where you combine some logical rules depending on your domain with one or more of these imputation methods. We also have methods that do multivariate or iterative imputation, where you can impute multiple variables at the same time. And so I talked about sort of the issue of doing multiple imputation, where, for example, if you are using random imputation, you can randomly draw different samples from a known distribution and then analyze all samples and then average the result from the imputed data set that you have, instead of using just one data set for inference. And so that is one advantage that multiple sort of imputation has over single imputation, that instead of using just one data set for inference, you can replace the missing value with multiple values. So you get, if you are interested in let's say imputing 10 data set, then you have 10 data set. You can then analyze each of the 10 data set and then average the result across the 10 data set and then use the pooled result as an inference. So for example, if your ultimate goal is to use the complete data set for just a regression, to fit a regression model to the complete data set, then you can impute the data set 5 times and then fit a regression model to each of the 5 data sets. You can then have an overall sort of a pooled regression estimate, which will be the average of the regression estimate across all 5 data sets. You can then calculate even the variance from this estimate by using the formula that you see here. And this overall, using multiple imputation, like I said, should be more accurate and precise than using sort of just one data set for inference. And so missing data is sort of very common in many domains, but it's particularly common in the biology domain. And so I think you've heard about different packages that can be used to sort of model biology data, which has missing data in it. One such package is the PMAT package in R, which was developed by people here in the lab. And the PMAT is an R package that can be used to analyze biological data in the presence of missing data. And it has many functions that helps with sort of processing the data. It has many functions which helps with sort of processing the data. It has functions that help sort of replace zero values with NAs. So often the left censored data set, which are 0 values, are are then replaced with NAs, so that if then there are functions in the PMAT that can sort of get rid of these, replace these zeros with NAs, and then the appropriate analysis can then be done. There are also functions to sort of transform the data set. You can do different transformation to sort of normalize your data set, the log, different kinds of log transformation, depending on the distribution of your data set. That can sort of then make you be able to then analyze your data set. There are also functions that I can use to sort of filter by molecules. So there are proteomics filter functions that then help you to sort of remove degenerate peptides based on based on some criteria, such as the number of samples, the number of peptides per proteins. There's also more molecule filter that can use to sort of remove by molecules not present in a certain number of samples. There's also filtering based on variation in your data set. So you can also sort of remove highly variable biomolecules. There are other custom filtering depending on your sort of criteria and then domain knowledge. And then there are other filtering that remove biomolecules for which sort of statistical analysis cannot be performed. There are also functions to sort of filter samples based on the Mahalanobis distance metric that is used to sort of remove outlier samples based on various sort of metrics. There are methods to also sort of normalize the data set where you choose a specific normalization strategy. You can use the statistical procedure for the analysis of peptides abundance normalization strategies, SPAMS, to evaluate and then choose from multiple normalization strategies to also normalize your data set. There are also functions to sort of summarize your data where you can display various correlation plot, like the correlation heat map. And then there are also multiple principal component methods like the sequential projection pursuit, principal component analysis, and then Bayesian principal component analysis that can be used to sort of summarize also your data set. And so this is how the output for summarizing your peptide data will typically look like using functions in PMAT. So it will give you the number of sample IDs in your data set. It will give you the number of unique proteins in your data set. It will also you can have some idea about the number of unique peptides in your data set, the total number of missingness in your data set, the proportion of missing in your data set, and then the number of samples per group. We talked about filtering, sample filtering. So often you have to identify samples which are sort of potential outliers or anomalies in your data set due to either preparation or any processing circumstances. And so you can use the Mahalanobis distance to sort of filter those samples out based on two or five of the following metrics. So either the correlation, Pearson correlation coefficient, the proportion of data missing across the sample, the median absolute deviation, MAD, the skewness of the data within the sample, and then the kurtosis of the data within also the sample. And so the Mahalanobis distance sort of measures the distance of each sample to the center point of your data set. And so potential outlier samples will be far from the center of your data set. And then these Mahalanobis metrics follow a chi-square distribution. So you can also calculate the p-values for them. And so in this case, the horizontal line here represent a p-value threshold where you can use that one as your filtering criteria. So any sample above this p-value can be classified as a potential outlier. So for example, in this case, you may be interested in sort of getting rid of this red sample here because of its high metric here. And this sample, you can look at based on these 5 metrics, it has a high proportion of missingness. And so it's also have a high sort of absolute deviation value. There are also sort of functions to also explore the pattern of missingness in your data set. So for example, you can have plot of the number of missingness versus your samples, where you can look at whether your missingness is uniform across the samples or not. So in this case, you can see the missingness is not uniform. There are samples that have high number of missingness, and there are samples that have low number of missingness. You can also explore the number of missingness versus your peptide intensity, where you can even then color it by other grouping variable by the strain and other even treatment group. So in this case, for this plot, you can see that the number of missingness seem to have an increment of 10. And so this could be due to either treatment effect or other variable that you can investigate also further. Okay, so some studies have sort of explored the relationship between missingness and then mean peptide intensity. And so these two plots from one study where they studied the relationship between missingness and then mean peptide intensity for human cluster and then lung data set. And for most of these studies, I think the conclusion was similar, where the relationship between the missing values and then the mean peptides was found to be negative. So there was a sort of a negative relationship between missing values and then the mean peptide intensity. The horizontal red line that you see here is represent a 50% threshold, where 50% of the peptide is missing across all samples. And then the vertical red line is the overall mean peptide intensity for the data set. And so, like I said, I think numerous studies have studied the relationship between peak intensity and the amount of missing data, and that they've shown that there is sort of a negative relationship between peak intensity and then the amount of missing data and the amount of missing values in the data set. And then this could be due to left censoring, where missingness is induced by low peptide intensity values. So under this assumption, it means that low abundant peptides should have more missing values, and then the fraction of missing values should also sort of increase as the peptide intensity decreases. But this is not uniform across all peptides, which means that not all peptides of low intensity have large amount of missing values, and likewise, not all highly abundant peptides have also high coverage. And so this means that the missingness that occurs here is a combination of not missing at random and then missing also at random. So in terms of the imputation method used in the biological domain, these imputation methods are sort of grouped under three main categories. We have imputation methods by a single decade replacement. And these are, for example, these methods include replacing missing with your limit of detection, and then replacing the missingness with half the minimum observed among all peptides, and also replacing the missing values with half the minimum for each peptide. There was also random till imputation. The other group is missingness based on local structures in your data set. And these methods include using the k-nearest methods, using the local least square method, and the regularized expectation maximization method, and other model-based imputation methods also. And then the last group include imputation based on your global structures in your data set. And these imputation methods include all the sort of the common principal component analysis methods, like the probabilistic principal component analysis and then the Bayesian principal component analysis. So then we've talked about a lot of sort of the evaluation. We've talked a lot about imputation method. So how do you evaluate? How do you know which imputation method to use? I think the simple answer is that the simple way to do this is that if there is a known truth, then you can sort of compare your result based on your imputed data to the known truth. So for example, if you have a data set that is created with some known dilution ratios, then you can compare the expected ratio to your actual ratio using things like the root mean square error, which calculates the mean square error between your predicted data and then your actual sort of data set. For experimental data, if you have two sets of samples and there is known truth, then you can sort of do the evaluation based on the classification accuracy, that is classification method applied to the analysis data set. So if there's no ground truth, then it becomes a difficult exercise. And then research in this is still open. And so what is typically done is that you apply different imputation methods, and then you then combine the result with sort of domain knowledge to choose which imputation method is best for your data set. Yeah, so yeah, that is all that I have for you today, and I'll be happy to answer any question that you may have.
We have an online question, Moses. How do you know which category the missingness belongs to? Is it MCAR or MAR or MNAR?
Yes, so that is a great question. So realistically, typically, you wouldn't know. You wouldn't know. That is why the first thing you do is try to do some data visualization, right? So try to sort of plot the missingness as a function of the other variable in your data set. Okay, and so, for example, with the survey data that we showed, if you plot the missingness in earnings as a function of education, and you see any pattern sort of coming up, then that may give you an indication that the missingness will be missing as random. But typically, you wouldn't know. You wouldn't know whether the missingness falls under any of the three categories. And so you combine data visualization with sort of domain knowledge to come out with sort of the best imputation method to use.
So for the multiple imputation, if you impute, say, 5 times, but every single imputation has the same bias, right? Of course, you would heavily skew the data. So how do you avoid, if you're using the same imputation? You said you randomized it, right? So assuming you're using the same imputation method with different numbers that you put in there, how do you avoid that? If you use different imputation methods, how do you know that they're like independent imputation methods to avoid that same problem?
Yes. So with the multiple imputation, is usually random imputation, right? And so if you are using, let's say you are using a regression imputation, right? You using a regression imputation means that you've determined the type of missingness that is in your data set. And then you've come to the conclusion that regression imputation is the sort of the right method to use, right? And so in that way, instead of just using one imputed data, which does not account for uncertainty in your data set, you can then sort of use random imputation where you are combining the predicted value with the prediction error. And like I said, this will ensure that you are accounting for estimation uncertainty in your sort of using just one value to represent your missing value.
Thank you.
Hi, Dr. Moses, a great talk. And yes, this is a real problem. I just I just wanted to know if there's like any I don't know, online resources that could help us make the decisions for our data set in this case, if that you know of.
In terms of determine which imputation method to use or the nature of missingness in your data set.
Come again?
Is it the online resources? Is it in terms of determining the nature of missingness in your data or knowing which imputation method to use?
Yeah, and to guide us through like which would be the options available for us to do the imputation in our data in this case.
Oh, yeah, I think there are various books available that go over different imputation methods. None come to mind right now. There is this paper by, I think, Lisa Bremer et al. That goes over different imputation methods that sort of looked at, evaluated different imputation method using, I think, in the biological domain. So I think I'll post some papers in the sort of chat, but there are papers out there that have sort of evaluated different imputation methods and in the biology domain. Yeah, so there are tools out there that can help you, tools that uses sort of data visualization as well.
Yeah, if you could share those with us in the Discord maybe later, that would be awesome. Thank you.
Yeah, I'll do that.
All right, we have another online question. If we have a sparse relative abundance data set, is there any better imputation method rather than changing them to 0?
Yeah, so with that, I would say that it will depend on a number of things, right? It will depend on your sample size. It will also depend on sort of the nature of missingness, right? And typically with sparse data, I think people argue for using imputation method that are used during sort of you model the missingness as part of your model estimation. And so, for example, the Bayesian approach is one popular method that people use when you have sort of sparse data set where you treat the missing value as, let's say, a latent variable. And so you model as part of your sort of data fitting, you model both the observed data set. You have a different model for the observed data set, and then you have a different sort of model for the unobserved data set. And so there are methods out there that can be used when you have sort of sparse data set. I think expectation maximization, for example, is known to do very, very well if you have a sparse data set and then you want to sort of model, you want to sort of estimate the missingness during model process. So like I said, you have two kinds of imputation. You can either impute the data set and then model the data set after imputation, or you can impute the data set during sort of model fitting. And so with sparse data, I think the better approach would be sort of imputing the data set during model fitting and using methods such as the Bayesian approach where you model the missingness as a latent variable, or using even expectation maximization where you sort of maximize the data set whilst you are sort of finding the parameter estimate, are also sort of using the parameter estimate that you are finding to also sort of estimate the missingness in your data set.
Yeah, I'd like to also emphasize a part of Moses's answer in that it really does depend on the missing data mechanism. So if you know, for example, that the NAs are a result of values being beneath that limit of detection for your instrument, you don't want to use a kind of imputation method that's ignorant of that. Zero imputation would make the most sense. Because otherwise, if you were to use something like mean imputation or a regression-based imputation, you'd be using observed values when you know, again, that the missing value is because it's beneath that instrument's detection level. So be aware of that missing data mechanism, and I guess first do that mental check of, is this because of my instrument that the data are missing, or is there some other reason that can explain it? So yeah. So another question that we have online is, are there any R packages you would recommend for these regression-based imputations?
In terms of packages in R?
Yes.
Yes, I think the popular package in R used for regression or multiple imputations is the MICE package. The MICE package has nice functions that can do both single and then multiple imputation. And it has a number of methods also there. I think it covers most of the method used in used for imputation. But for omics or biological data, I think the PMAX package also has sort of great functions that can handle missing data well. And so I would say using both the MICE package and then the PMAX package should be sufficient there.
Okay, another question. What is the difference between the imputation p-value and the traditional p-value?
In terms of the Mahalanobis distance metric, so with the traditional p-values, technically, I think they're all the same. With the traditional p-values, you are comparing some test statistic to some threshold of a distribution, typically a normal distribution, right? And so you calculate your effect, and then you calculate the test statistic, and then you are compare that test statistic to some value from, say, a standard normal distribution, right? So it's the same thing that we are doing here with the Mahalanobis distance. Those metrics are assumed to have a chi-square distribution, right? So once you calculate it, you then compare those values to a chi-square distribution, and depending on where they fall, right, that value there, that depending on where they fall, you can then calculate the p-value based on that. And so technically they're all the same. You are comparing some calculated value to some value from a distribution, in this case from a chi-square distribution.